our group, Alexis, Clayton, myself, and Ray, we studied the question, does Echinacea extract halt the development of the mosquito Aedes aegypti development? First, we'd like to acknowledge Dr. Kolinsky and Professor Nickel for helping us come up with the idea for our lab and helping us with statistical analysis. We'd like to thank Melissa, who's no longer in our group, but who helped us collect data. We'd like to thank the William Jewell College and Biology Department, the Biology Department for providing funds, and the Physics Department for providing space for our experiment. We'd also like to thank Campus Safety for letting us into the building late at night. So there are thousands of different diseases, all of which are deleterious in our health. And some of these, roughly 17%, are considered vector-borne, which means they're spread by an organism from human to human or potentially from animal to human. One of the most prevalent of these vectors is the mosquito, which I'm sure we've all heard of, and it's known to transmit diseases like dengue, malaria, yellow fever, and Zika virus. You've probably all heard of more recent escalation of Zika as it spread through through South America, has caused national health crisis in countries like Brazil, and its association with diseases or malformations like microcephaly, shrunken headness of fetuses, has caused some great concern. And recently, it's been noted that it's spread somewhat into North America, which makes it very prevalent, and it's in the same family as the dengue virus, and both of which are spread specifically by the mosquito species Aedes aegypti.
Aedes aegypti are found in humid and warm areas in places in the United States like Florida, where Zika has spread. Females lay their eggs in water, and these bodies of water can be as large as a pond or as small as a pool, water pooled in a tire or a trash can. Females lay 100 eggs at a time, and these eggs go through 4 stages during their life. The eggs, larvae, pupa, and adult. And the reason why Aedes aegypti are so dangerous is because they thrive in urbanized areas like Florida.
So one of the ways that we can solve the problems of disease spreading is through insecticides. And so insecticides can function in many different ways. They can be toxic, they can affect the development, and they can also affect behavior. So toxic insecticides just kill the insect outright. Behavioral, instead of killing it, it'll simply modify the behavior. And then developmental will inhibit the growth of insects. And so an effective way of thinking about how developmental insecticides work is through an analogy. So if you're ever driving on the main road to get to a destination, oftentimes there are stoplights along the way that are inhibiting your ability to get to that destination. And so in insects, it works very similarly through a hormone called juvenile hormone. And any time that the juvenile hormone is present in the insect, it'll be like a red light, and the insect won't be allowed to develop into an adult. And so developmental insecticides work by mimicking juvenile hormone and keeping it constantly in the insect and preventing it from developing into an adult.
So research on juvenile hormones is relatively new, but we did find, well, relatively few and far between is a better word, but we did find a very interesting paper written by Jacobson et al. In 1975, where they took the plant Echinacea and extracted a compound called equinolone from the plant's roots. Equinolone works as a juvenile hormone mimic in yellow mealworms in their research, where it halted the development of mealworms from the pupal to the adult stage. So basically, most of the individuals died in the pupal stage rather than making it into adulthood. And that gave us inspiration for our own question and hypothesis, where we were talking about mosquitoes. So we were looking at whether or not Echinacea had a probability of halting development in mosquitoes, and we hypothesized that Echinacea extract would halt the development in mosquitoes between the larval and pupal stage, and also between the pupal and adult stage.
So in order to set up our experiment, we needed to create a suitable environment for these mosquitoes to survive, and that needed to be warm and humid. So to start, we put insulation on a table, and then on top of that table we put heat tape. And if you notice on the right side of the heat tape, there are wires, and those wires connect to a control panel, and you can turn the heat up and down. They didn't really have numbers or anything, but you just kind of had to gauge it. We had several trial and error. So then we put an aquarium on top of the heat tape, and then to initiate the hatching phase, we place a strip of 1,000 eggs into a tub of warm water, And once those eggs hatched, we individually picked up the larvae and placed them in cups, and then we placed the cups inside the aquarium.
So to test the effect of concentration of echinacea extract, we had 10 replications with 3 different one control do 2 different concentrations. Our control received no echinacea. Our 1X treatment received 0.5 milliliters in the 30 milliliter cups, and the 2X received 1 milliliter of echinacea extract. And in each one of these cups, we put 10 larvae, and we had 10 of each. So for a total of 300 mosquitoes we tested.
So once we had the experiment set up, the experiment ran for 16 days. We fed the larva every other day. We took temperatures every day to try to keep that as constant as we could. And we took measurements of the number of pupa, larvae, and adults every other day and recorded that on a spreadsheet. Every time we found a dead specimen, no matter what the stage was, we removed it and we preserved it in ethanol to kind of keep the counts as relevant and accurate as we could keep them.
We analyzed our data using a chi-square goodness of fit test, and a chi-square test tests how closely data resemble expected values. So you go into a chi-square test expecting there to be no correlation with independent variable have no effect. And so by summing the differences between the observed and expected data, if we get a large number that correlates with a p-value of less than 0.05, then we can say that our set of data is statistically different than no correlation. And so if our hypothesis was correct, we'd expect to see results that look like this. On the y-axis, we graphed how many individuals got to that particular stage but no further. And so in the control treatment with no echinacea, we would expect many of the individuals to reach the adult stage. In the 1X treatment, we would expect some of those individuals to not make it past the larval or pupal stage, and in 2X, even fewer individuals make it to adult stage.
So those are expected results, but this is what we found. So as you can see, they're quite different. You can notice on the y-axis we have observed frequencies, and that's observed frequencies of individuals who only made it to this stage and didn't progress. And then on the x-axis we have treatments. We have the control 1X and 2X. The bars represent, the dark gray bar represents individuals who made it to the larval stage. The medium gray bar represents individuals who made it to the pupal stage, and the light gray bar represents individuals who made it all the way to the adult stage. And you can see that there's letters above the data. That represents the statistics that we found when we did our analysis. So we found that when we did the chi-square test on all of the data together, we found that the control and 2X were not significantly different, but the control and 2X were significantly different from the 1X. And that p-value was 0.003.
So our results were significant with a p-value of 0.0003. Even though our results were significant, we still reject our hypothesis because it cannot be supported that echinacea extracts alter the development of Aedes aegypti.
That's mostly because of the results that we saw in our control group. Because most of our control group died, we can't tell you that echinacea was actually responsible for halting the development because we saw pretty much the same results across the board.
So some of the biological reasons have to do with our methodological errors that we performed. So one of them, the temperature of our cups was supposed to be maintained according to literature around 25 to 29 degrees Celsius for ideal development. Our temperatures reached up to 32 degrees Celsius, which in part is due to the volatility of the heating tape. There was also the issue of feeding. We weren't exactly sure how to feed because all the literature we read pretty much stated giving them pinches of food, which isn't very accurate or precise measure, I should say. And also when we called the company to ask them exactly what we were feeding them, they didn't know what was in the food exactly. We knew it was mosquito food. There was also the issue of the tap water. For the first couple days, we grew the larvae in tap water without thinking about it. And we did switch it to deionized water shortly after. But there could have been ions, chlorine, other non-ideal elements in the water that could have halted or altered the development of mosquitoes.
So essentially, we didn't know how to raise the mosquitoes going in, and so we could correct this by actually running pre-experimental trials, where we would take a portion of the eggs that we purchased and raise them in our habitat and test for the best way to regulate temperature, best way to feed them, so that we could ensure that we have a better baseline control to compare our data to when we actually run our experiment.
Overall, this still is very important research. Research in larvicides is important because we need like disease spreading across the world. And research on echinacea is still very important because literature all the way from 1975 to 2015 advocates that echinacea might possibly have mosquitocidal properties. So it's really important that we continue on with this research because it could be a very probable solution, one of the solutions of many to solving problems like worldwide spreading diseases. Here's our literature. And questions?
So how would you propose using this extract in real life?
Our idea was if we could find that putting extract in water could halt the development of mosquitoes, like you could maybe put it in the small gatherings of water that are around your house, like maybe puddles, where there's just a little bit of water that mosquitoes could possibly develop in, and it could be kind of like a home remedy idea, because you can buy it at Walmart,
Maybe I missed this, but you guys talked about a form of beetles, right, that used that was halted.
Research, yeah.
Yeah.
They halted.
Did you guys have any research that said there was any link to mosquitoes, or was it kind of just guessing?
They're both holometabolous species,
Okay.
meaning that they have 4 distinct life cycles from egg, larvae, pupa, and adult.
Okay.
And so we figured because of this, both these types of, by the way, these types of species have these hormone fluctuations as they're developing. And so if this juvenile hormone that is supposedly present in all these different species is found in echinacea, specifically at the concentrations we used, we should be able to detect a difference in development to adult stage.
But if I can add to that, on our last slide, our big picture slide, this Manetti et al. 2015 was kind of a rundown of echinacea in general. It was kind of like a detailed analysis of the different potentials of echinacea and the different ways that echinacea can be used, and it advocated that echinacea might be have mosquitocidal properties for Aedes aegypti specifically. So it was really interesting and kind of added to our notion of further research.
Sorry.
Do you know what it is about echinacea that mimics juvenile hormone?
Specifically the compound equinolone, and we wanted originally to synthesize that and have it in its purest form, but it's super complicated and very difficult. So we ended up just using the extract in general to see if we could get the same results as they did in Jacobson et al. When they used the kind of synthesized compound.
And we don't know exactly the molecular fittings into receptors. The juvenile hormone receptors are still being researched. Papers I've read suggest that there's not a ton known about the juvenile hormone receptors. Maybe there's been updated, but as far as we know, we just know that there's a quinolone that supposedly acts as a mimic of juvenile hormone, and that quinolone was extracted from echinacea, so we figured it should be in the concentrated echinacea extract that we use.
So between the beetles and the mosquitoes, what leads you to believe that it's the same juvenile hormone that it is?
The paper I read referred to juvenile hormone amongst holometabolous species as a constant hormone.
That's all the same chemical hormone?
Yeah. That's what
Okay. So when you originally transferred the eggs to the cups, the mosquito cups, were they already hatched or had they not hatched?
Yeah, they were already.
They were already hatched, yeah.
Yeah.
Okay. Why not put them in the cups before they hatch and see if juvenile hormone could stop them from hatching?
Well, the eggs are pretty infinitesimal, and they're on a strip, that kind of a paper strip that's,
Okay.
it would be kind of hard to remove them from the strip and
Okay.
count them all out. And we wanted to make sure that some of them didn't hatch at all, so we wanted to make sure that we were actually getting 10 in every cup.
That also wasn't our experiment, neither. That wasn't supported by previous research that we looked up. The idea of halting the hatching of eggs is a different set of hormones, and it would be a different study to be investigated. It should be investigated, and there could be potential there, but I don't know about echinacea extract being used for that. I'm not sure.
So with the juvenilizing hormone, it sort of traps them at a stage of development. What happens to an
So in Jacobson et al., the pupae kind of lived for a few days and then it eventually died, and that's kind of like what we saw in our research. I mean, not that we got any acceptable results, but I assume that they can't just live forever in the water in that way.
So I read that the tissues that are in the larval and the pupal stage, the soft tissues, aren't made to be around long term because they're supposed to develop into adults. So that's what happens is the tissues kind of break down and they insect dies.
All right. You knew.
Could you go back to your graphic that shows the different types of insecticides? While you're doing that, I'm going to ask a different question. You mentioned that Aedes aegypti thrive in urban areas, and I just wondered why they would thrive in urban areas compared to rural.
There is, I found literature, there's subspecies of Aedes aegypti, specifically Aedes aegypti, I believe they were referred to. That's the subspecies. They comparatively selectively feed on humans, and they actually oviposit selectively on human containers, just because comparatively to a different subspecies, I don't remember the scientific name, who oviposit, lay eggs in tree holes and stuff like that. They compared these two subspecies, and they found that there are, in fact, definitely certain subspecies that prefer to live, feed, and lay eggs in human environments, which means that anywhere humans go is a hospitable environment for these mosquitoes to thrive, which makes them a particular threat for spreading disease within a population.
Okay, I have a question about this slide. I'm curious, so what would you say is the definition of an insecticide? Like what is an insecticide? What should it be? What should it do?
So the side suffix, if I understand that, means death,
Right.
to die. So toxic, that the goal is to kill the insect. Developmental kills it just in a sort of a different indirect way, and then behavioral doesn't necessarily have to kill it. So the example that I thought of was how bee harvesters smoke out beehives to subdue them to get the honey out. I think Dr. Quincy also taught us about introducing a bunch of sterile males into a population to kind of lower the reproductive rate and growth. So
this
I
makes me
kind of thought of
die.
insecticides as far as like how we're using them. We're trying to we want some kind of advantage, whether it be like crops or spreading disease. And so we just alter the life cycle or how insects behave in order to benefit us.
It wasn't just totally clear from the slide that all of these ways can result in death. Basically, you're just trying to sort of so I just wanted to
Yeah.
Yeah, stromal technique wouldn't actually be classified as an insecticide. It's a different class of ways of controlling insects. And was actually one of the questions I was going to ask you, which was, what are other ways of controlling insects other than insecticides? And you've started answering that already, so I won't belabor that point. So insecticides are almost always chemicals of some type. So what would be a way in which a chemical can disrupt behavior? Just to follow up on Professor Nichols' question. Did you read about any specific examples?
I don't know if, do you accept the honeybee example, or is that, no, that's not a,
That's not doing anything but making
to
them
make them behave differently?
making them behave differently so you can go in and do something. As soon as you remove that, they're going to go back to doing what they were doing before, which is producing a ton of bees and honey and everything else.
Right.
What kinds of behaviors do these behavioral disruptors usually affect?
From what I read, there was some reproduction effect, and
I'm thinking pheromones in reproduction.
Yeah, they can also oftentimes either mimic pheromones or they can actually disrupt the individual's ability to detect pheromones. We talk about pheromones being used as as attractants for moths and stuff in class, a lot of times they disrupt the chemical communication among insects because communication is really important. Aaron, you refer to mosquitoes as tree hole breeding insects. Describe a little more what you mean by that.
There are certainly subspecies, even within Aedes aegypti, that breed within tree holes in any small pockets of water where there is.
How small?
As small as a tree hole. I don't know measurements.
As small as a soda cap. It could be.
Sure.
Could be.
So in your experimental setup, why did you choose the size of containers to put these larvae in?
Mostly because the fact that we weren't going to be replacing water, like taking water in and out, means that we probably needed more water so that scum wouldn't build up from like the food. Because a lot of the studies that we read said that scum built up over time with food, and some of the studies even did remove water, but we thought that would be complicated with our treatment, keeping the ratio correct. So we thought a little more water might reduce the chances of a scum slime buildup at the top.
Go to your experimental setup. Are those containers when we see them in a minute?
I'm sorry.
That's all right.
We have a million things.
Are those big containers or little containers?
They're little.
I mean, they're big.
I think of them as little containers, actually. So, did the fact that this is a tree hole breeding mosquito make your life easier or more difficult in the process of doing this whole thing?
Probably easier. I mean, it was nice that we kind of had, a very concise, small setup here.
It was more convenient to set it up like this, but managing them and managing this, because there was scum buildup, and there was issues with, just having them in such small containers made it difficult because other literature referred to rearing them in pans, but because we wanted to test concentrations in various sections, we had to split it up in small containers.
So, a lot of times when you guys finish projects like this, you go into troubleshooting mode, like, oh, what could we have done differently? And you mentioned the fluctuations in temperature. And I could have told you this back at the time. You can leave that there. Leave that up there. I could have told you this at the time when I thought about it, but I didn't actually think about it until just now. What's one thing you could have done that might have helped you with your temperature fluctuation issue, other than being in a building that doesn't just fluctuate radically in temperature from day to day?
Literature that I read suggested things as simple as a heat lamp. I don't know that really would have helped because I feel like heat tape is relatively just as kind of variable as a heat lamp, but and things as complicated as a temperature and humidity preset kind of
Incubator.
area.
That would have helped, but we didn't have access. Freedom to use one.
Yeah, I thought as I was looking at that, you could have put a 1/4 of an inch of water across the bottom of the tank. Water heats up and cools down slower, and so when the room did fluctuate, having it kind of in a water bath like you would in a chemistry class, we all used water baths in the past. It's like, why did it take me this long to figure this out? Just put that in your bonnet for any other time that you need to control temperature this way.
Yeah.
You might find
Yeah.
yourself needing to control temperature a little more precisely, and a water bath would help.
So you felt like the temperature got too warm?
According to some of the literature we were reading, they recommended, like I said, 25 to 29.
Wasn't there a time period when it got too cold?
It did.
Yeah.
There was a too cold. There was a lot of
There's
issues.
a lot of issues
but
with the range.
overall, we started to move them away from where they were exposed because they were getting too hot around here, and then they would get too warm in the center, and so we redid the arrangement every single day because we were like, they're too hot here. We're gonna try to push them to the cooler areas.
And how did you know that they were getting too hot?
We took temperatures every day, and
How did you do that? Physically?
with the thermometer in the hole that we made in the lid.
Yeah.
So,
Yeah.
and then we would,
Yeah.
go to your results slide with your graph. Your bar. So you did well in describing kind of the statistics here and saying, I understand that A's are so you have significant difference, and the A's are the same and different from B. So what? It's hard to tell from looking at the graph though. What is different?
Basically we got the statistical difference from B because even though it doesn't look like a lot of difference, this is like 14 individuals who overall made it out of the larval stage, whereas A is only 4 and this one is 0. So like even though it doesn't seem like a huge difference, it was different enough to make a statistical difference, and it was just basically like the number who made it out of the larval stage.
And in that 2X treatment, so all of them died. Was that immediate?
It was much faster than the other treatments.
By day 6.
6 to 8 maybe. And the other treatments, I mean, we were counting 1X treatment until day 16, and so.
Why do you think that was?
The happy, like, maybe part of me wants to say that if our controls would have acted the way that we wanted them to act, maybe it was because the echinacea was actually having an effect, you know? And so, like, these groups might have been what we would have seen if our hypothesis was upheld more, but since our control group is such a mess, you know, we can't.
There's, as she said, there's research suggesting that there are mosquitocidal tendencies of echinacea extract, dealing with certain chemical alchemides, I believe, that actually are known to kill developing mosquitoes at high concentrations. So, had like she said, had our control worked, we could have maybe come up with
It might have been because
some
our
results.
hypothesis was supported, but I mean, realistically, it could also have been because,
scum buildup from echinacea.
or you know, many of the other methodological errors that also killed the control group relatively early. You know.
Alan Kovich, a faculty member at the University of Georgia, once told one of my students that in 40 years of doing science, he never did an experiment that worked the first time he did it. With that in mind, there were probably also plenty of experiments that he tried the first time and then decided to never try again. Is this something that might be worth revisiting after you think about some of your methodological difficulties, or do you think this is just a terrible idea?
I think it's worth pursuing in the future because mosquitoes spread very harmful diseases to humans, and in my lab report I stated that mosquitoes are one of the #1 causes of deaths to humans via malaria and Zika. If we were to reduce the number of adults who in the world, then we could potentially reduce the number of deaths per year.
Also, I just think it's a cool idea. I mean, like, not a lot of research has been done on it. There's no, like, definitive answer. And in Bio 135, when we were reading kind of the journals of Wilson, it was like, you know, pick something that isn't super well studied, that's kind of out in the field, and just go for it. And I would go for it again. I think it's really interesting, and there's still a lot of potential. So
There should be more research on any negative side effects as well, because since it's not a very looked into method, more research, it warrants more research if it works like Equinolone did, and then it's a trade-off between safety and effectiveness should be looked at as well.
I'm done whenever you are. But you have another question back here.
What do you think, why do you see the need for echinacea? Like, what's wrong with the other insecticides that could be used for this purpose?
one of the biggest problems is that when you have these like widely spread chemical and like insecticides that are like being sprayed over plant populations, a lot of plants develop a resistance over time, and they can kind of like selectively develop resistance to a lot of the literature that I read said that like because we only have a few types of like mainstream insecticides really, that resistance can be developed, and for that reason you need like a variety of, you need your hands in a lot of baskets to be able to like fully combat the issue.
So what happens when they develop a resistance to echinacea?
I mean,
Find
of
something
course,
else?
we will always
Yeah,
be like,
that's-
Just
I mean,
constantly
same
finding
with
something else.
harmful bacteria.
Yeah,
There's
you'll-
always resistance and more research on you. But if we can understand if these types of extracts in themselves can act like mimics, then there is potential for more research in other plants and other types.
If something mimics a hormone that the organism produces, what are the odds of that species becoming resistant to that particular intervention?
I would, oh, go ahead. Oh, I would say, I mean, like, because it's not necessarily like foreign, I would, in my own, just kind of thoughts, I obviously haven't done any research about this, but in my own thoughts, because it's not a foreign kind of body and it's something that they already make, I would say it's less likely.
Imagine if mosquitoes developed immunity to their own juvenile hormone. Then there would be unregulated growth. And when they do tests where they try and limit the amount of juvenile hormone, they die as larvae. It's in homology. Not necessarily mosquitoes, but other insects that use this mechanism.
When they knock it out.
Yes, not gene knockout, but hormone receptor protein.